look at the second part of the Chapter 2 lecture on cellular adaptations and injury. So when we talk about cell injury, there are two what we refer to as reversible types, things that damage cells and cause cellular swelling. So as cells start to fill with fluid or substances and they swell, and there's what we refer to as fatty change, where lipids can accumulate in the cell. So in both these cases, they are something that we can recover from. Our cells can recover. Fatty change is more difficult to recover from, but it is still reversible. In terms of cell death themselves, we'll talk a little bit more about these in a few minutes, but there are basically two categories of cell death. Necroses. Necrosis is always pathological. It generally elicits an inflammatory response. So necrosis, always, when I say pathological, I mean it is always abnormal. It's almost always associated with inflammation, and therefore it leads to cell death. Gangrene is something else we'll talk about that goes with that later. Apoptosis is a selective process. It may be normal. For instance, as your cells normally age, they start to break down and your cells send a signal, hey, the cell's getting old, and they self-destruct. We recycle the parts, build a new cell. It's a normal process. Now, there are some abnormal things, some injury that can trigger apoptosis, but it's not the same as necrosis. First of all, necrosis never involves inflammation. Even when it's triggered by something else, it is not an inflammatory response. And then there are two pathways. And your book goes into a lot of detail on this, and I'm not asking you to know this detail. What you need to know about apoptosis, I'm covering right here. So understand that it is a selective process, often comes with older cells. It can be triggered by some kind of injury, but it does not involve inflammation. The extrinsic pathway is that the signal for apoptosis comes from outside the cell. And that's all I need you to know about that. It's an extracellular signal. Something outside is causing the problem. In an intrinsic pathway, the signal for apoptosis comes from inside the cell, and it is generally triggered from the mitochondria. And that usually is due to some sort of loss of oxygen or some kind of damage to the DNA. So intrinsic, internal cells. That doesn't mean this. It does not mean that injuries can't cause that. It just means that the pathway that leads to the process of cell death comes from inside the cell. Extrinsic means something outside is triggering the process of apoptosis. Now, when we look at the cellular responses in a general sense, there are several things that figure into how severe the injury is going to be. What are we going to see happening to cell injury? Is it going to lead to death or not? And so factors that figure into that are things like the nature of the injury. So what is it that happened? For instance, a burn can be more severe than, say, loss of nutrition to the cell. Lack of oxygen can be far more severe than, say, certain chemical exposures. Some chemical exposures take a lot longer, so that's the duration. So we have what is it? Some injuries are more severe just by the nature of what they are. The length of exposure, so very quick exposure to radiation, for instance, might not be as severe as a longer exposure to lack of oxygen. And then the severity is really, is it a large amount or a small amount? So you have the nature, what kind of injury is it? What is the thing that's causing it? How long does it occur? And then is it a severe or is it a mild exposure? So sometimes there are different types of radiation, and we'll talk about them in a minute. And in the more severe types of radiation, even a small amount can be really bad. Whereas the more mild radiation, like what you get from laying out in the sun, can possibly cause sun damage to your skin, but it takes a lot longer, so the severity of the injury is usually much less. When we say type and state of cell, we're really talking about, how specialized or differential is the cell. Cardiac muscle and neurons are far more specialized than epithelial tissue. And then adaptive processes that the cell can endure. There are some cells that can adjust. Epithelial cells can adjust to things. So those things figure into how cells will respond to injury. How well the cell's tissue organ, the person recovers depends upon their age. Young and old are more vulnerable. Are they well fed? Do you have good nutritional status? That helps enormously. So if there's starvation, poor eating habits just in general, the elderly tend to have poor eating habits, especially those who are on fixed incomes, live alone, eat a lot of canned soup, those kinds of things. They're much more vulnerable than someone who's eating lots of fruits and vegetables and calcium-rich foods and so forth. And then their overall health. How's their immune system? Are they already suffering from other diseases? That can make it a lot harder to recover. All right, so mechanisms of cell injury. Before we look at specific things that cause cell injury, like specific chemicals or radiation or so forth, we have to first look at what the mechanisms are. And there are three mechanisms we're going to go through. We start with free radicals. So first it says, well, what is the source of a free radical? And then I'll explain what I mean by a free radical and how and what they do. Free radicals can come from radiation. That's the primary damage that radiation causes, is they form free radicals. If you'll think back to our lecture on metabolism and the electron transport system, the ETS, remember along the cristae of the mitochondria, those electrons were captured, and then I said they were neutralized when they came in contact with hydrogen and oxygen to form water. When I said they were being neutralized, what I basically mean is they had the potential to become free radicals, but we kept them secure in the membrane and then helped neutralize, not let them run off free by having them bind there to form the water. So definitely, though, if that doesn't work right, there's processes of metabolism that can lead to, especially if there's lack of oxygen, so there's no oxygen or hydrogen to make the water. If the oxygen levels run low at the tissues, then that's going to be a problem. That'll lead to free radicals. So what is a free radical? Really, what we're talking about is an electrically charged atom or group of atoms that is unstable due to an unpaired electron in its outer orbit. So here you go. You got to think a little chemistry now. Remember what an atom looks like. It's got protons and neutrons in its nucleus, and then it has those shells of electrons spinning around it, and the number of shells, and remember the different number of shells or different number of electrons per shell is set, but how many shells they have depends upon the nature of the charge of the electron, how many protons there are, and then that are positively charged, and the electrons then balance that out. So it's possible for certain atoms to become unnaturally charged. In other words, they have an unpaired electron or an X. Sometimes it's an extra, sometimes they've lost one, sometimes they've gained one. Either way, they're no longer a neutral atom. They have this unpaired negative charge. So that's a problem. So when you see that not every molecule that has a negative charge is a free radical. We see that a lot with what we call superoxide, it's really an O2 negative. It's a single oxygen molecule. Normally we see, when I say O2, I don't mean oxygen O2. I mean I can't write on my screen right now, sorry, an oxygen with a 2 negative charge. In other words, it's not O2 with a subset 2. It's 2 with a negative. The oxygen, a single oxygen with a negative 2 electrons. That's unnatural. We usually see 2 oxygens bound together. So that usually comes, by the way, the superoxide free radical comes a lot from problems with metabolism. So what are they really doing now? They've got this unpaired electron and they're trying to get rid of it. They're trying to neutralize themselves. And so what do? And our picture kind of shows this. Those free radicals, those electrons, are being thrown all over the place in an attempt to try and get rid of it. They try and bind it to other things. And so what do? They damage things because they do that. For instance, here's our cell membrane. Those phospholipids are spots where, in fact, an electron can bounce out. Say if the mitochondria did have a metabolism issue going on, lack of oxygen, then the electrons can bind to the phospholipid membrane, start poking holes in it. As soon as you start poking holes in cells don't behave very well anymore. They'll swell And if you don't fix the holes, because when they swell, things start to flow inside the cell. The whole integrity of the cell is lost. They'll alter proteins. So when you look at the proteins, whether it's in the cell membrane or somewhere in the cell or even in the extracellular space, wherever there is proteins, they get to the proteins and they denature them. Remember what a protein is. It's a string of amino acids, and it folds once and then it folds again for a tertiary and then a quadrifold, so that you have 4 different folds that make that very complex protein. Those bonds that allow the amino acids to fold in on each other to make that protein, they break apart with an electron. And then it can get into the nucleus and it can alter the DNA. So once you do that, now you start forming abnormal proteins, abnormal cell functions. And when we start forming abnormal things, we want to start thinking about cancers that form. So free radicals are associated with cancer formation. So particularly on this third one, when they alter the DNA. So how do you protect or prevent free radicals? Well, most healthy cells have their own mechanisms to neutralize them. So there's lots of ways to protect them. And vitamins, vitamin E, vitamin A, and vitamin C are really effective at neutralizing free radicals, breaking the reactions they start, and making things better. So don't think, oh gosh, we have free radicals bouncing around, there's nothing we can do about them. No, in many cases we're safe. We've got them forming and we've got ways to neutralize them. The second mechanism of cell injury is impaired calcium homeostasis. And with impaired calcium homeostasis, what we're really talking about is really an intracellular, so we're looking at inside the cell, an intracellular calcium influx. So now we've got calcium starting to move into the cell. On our cellular adaptations and injuries earlier, part 1, we talked about calcium accumulating extracellularly. So it can get on heart valves, it can get in blood vessels, and this can cause some adaptations and possibly even some injury. But when we talk about calcium homeostasis here, I'm talking about inside the cells. We should not have hardly any calcium free floating around inside our cells. Because it's a mineral, it can start to conglomerate together, calcify together, and cause problems. So, but there's types of injuries that can lead to impaired calcium homeostasis. So, if this should happen, if the calcium ions start to increase inside the cell, especially in comparison to outside the cell, it damages the phospholipid bilayer. It damages the cytoskeleton because it starts to accumulate and the cells become rigid. It damages membrane proteins. So again, that's part of that phospholipid layer problem. So it's hitting the phospholipids, it's hitting the proteins in there. And then it increases the use of ATPase. Remember, that's the enzyme that breaks down ATP. So what that does is by increasing that use, it depletes our ATP stores, and so the cells lack energy for function. And then the third mechanism of cell injury is hypoxia. And you'll see I have this quote here, all roads lead to hypoxia. Arguably, if I go to free radicals, and with the damage to the free radicals, it can affect the cell membrane, and in fact can lead to fluids inside the cell, and hypoxia can be involved there. Hypoxia can also form free radicals. So there's links of hypoxia and free radicals. Theoretically, when we start to increase the ATPase and the depleted ATP, the cells will use up whatever oxygen it can to try and make whatever it can, and the cells can start to suffer from lack of oxygen as well. So these really are separate, but I could lead a pathway in which many things lead to hypoxia. And those are just in the mechanisms. When I start to talk about the kinds of injuries that happen to our body, whether it's at the tissue level, the organ level, or the full body level, we're talking about hypoxia. So lots of things cause that. So I'm not just talking, by the way, here about, when we say hypoxia, we're not just saying, okay, I fell in the pool and my lungs are filling with water and I'm drowning. That's hypoxic conditions of the whole body. But there are areas in the body where a blood vessel can be blocked. And I block a blood vessel that's carrying oxygenated blood, so now no oxygen gets to that tissue, and that tissue becomes hypoxic. So hypoxia can occur from a lot of different things And again, as we start looking at other diseases, we look at all of our cardiovascular diseases, they're all really going to lead to hypoxic type injuries. You look at respiratory, certainly that's the case. Even cancers can deplete cells of oxygen and therefore lead to hypoxic conditions. So lots of things, and here's some of the things listed here that can do that. Now, the bottom line of hypoxia is that we run out of ATP. When we run out of ATP, we can't do any functions for the cells. When we can't run things in the cells, everything falls apart. This is a little different than the description in the book, but I'd like you to know these steps in order as they are here. So these are really the manifestations of what really starts to happen with hypoxia. Now, if I look at it from a tissue level, by the way, one of the earliest things that you may see is a change in color. So if I have hypoxic tissue, the pinky kind of normal color will go to kind of a pale to blue wherever the oxygen is being deprived. Lots of hypoxic conditions are not visible, so I don't have that as a step in there, that loss of color. But know that that really is happening. As tissue loses oxygen, the color of the tissue will change. But really the first thing that you'll see happening in organs that are in tissues that are involved is any muscle that's involved will start to have difficulty with contraction. This is due to the reduced amount of ATP that's happening, because when I don't have oxygen, I can't do anything more than glycolysis, right? I need oxygen to do Krebs, I need oxygen for the ETS. So if I cannot get those two things done, I'm left to rely on glycolysis. I'll go from pyruvic acid to lactic acid, and I'll get a couple of ATP as opposed to the 36 or so I was going to get from the full process. So we have loss of muscle contractions. And again, it can be smooth muscle, it can be skeletal muscle, it can be cardiac muscle, but pretty much every area in the body's got some muscle walls of GI tract and so forth. They're all smooth muscle lines. So anywhere there's muscle, that's going to be the first thing you see starting to happen. Because the cells, the tissue are not able to go through that Krebs and ETS, the use of mitochondria diminishes, and they actually start losing them. The cell says, okay, whoa, let's grab everything I can from that mitochondria cell membrane, any nutrients I can, anything I can use at all, and so they start breaking down the mitochondria. Any glucose that was left, any glycogen stores that were left, anywhere they can grab it from, the cells are trying to grab it, and they'll deplete any storage that's been left inside the cells. No place else to get glucose, because you're using every bit of it just to go through the simple process of glycolysis, and you're only making about 4 ATP per molecule, so you're going to use that up really fast. At that point, now you're really out of ATP, and your sodium-potassium pumps for your cells start to fail. Those require ATP to function. They're maintaining the cell integrity, and as soon as they fail, you hit what we refer to as the point of no return. At this point, you can get oxygen in right here, but if you don't bring in oxygen at this point, the odds are you will never recover. And I'm not talking necessarily always whole body. Remember, we're talking about a particular tissue. It may be the whole body, but it could just be one particular area of the body. But this is sort of our point of no return. Now, we make strides in medicine every day, and certainly every year that I teach this course. And so it is true that there's been some research out there that in hypoxic conditions, there's been some improvement. And so we've actually had a few cases where it's gotten even further than this, and tissue has been recovered. But for the most part, I want you to be thinking about this as pretty much it. If I don't introduce oxygen now, I'm not going to be able to get it back in. If I'm putting it in after this next step, I'll just do more damage than good. And so this next step is referred to as vacuolation. Vacuolation is actually when you get, kind of looks like bubbles forming inside the cell. If you look inside the cell, hold on, I'm sorry, I'm at home and all right, sorry about that. So vacuolation is what we were talking about. So vacuolation is when the swelling worsens, and again, if you look inside the cell, it looks like there are bubbles forming. They're actually temporary membranes that are forming around bits and pieces of the cytoplasm and the components of the cellular organelles. So if you look inside a cell, that's in vacuolation, it looks like there's bubbles inside the cell. So this damage now starts breaking apart the cells. Soon those bubbles will start to affect the function of the lysosomes. The lysosomes won't be able to break down any of the damaged parts of the cell. They start releasing enzymes all over the place and the cell will die. So vacuolation, again, it looks sort of like bubbles inside the cell if you look at it, but they're actually membranes that start to form around components of the cells in cytoplasm. It's like little chunks of it, like the cell is breaking into chunks. It's really a protective mechanism of the cell to try and manage what's happening, but instead it damages the lysosomes, enzymes are released, and the cell is destroyed. Now, reperfusion injury is what occurs if oxygen is reintroduced after this point of no return. So once vacuolation occurs and then you put oxygen back to the tissues, you actually can start forming those oxygen molecules into free radicals, and then you make the damage even worse. So again, I mentioned before, there have been some cases in some extreme hypoxic cases where they have actually been able to save some tissue, but by and large, once vacuolation occurs, you can no longer save the cells, the tissues, the organ. It's lost. The cells will now die. All right, so those were the mechanisms of cell injury. So what kinds of things cause cell injury? These are the things that I want you to know about. There's a lot of other ones in the cell, in the textbook. We're not going through them all. But in terms of the causes of cell injury, we start with radiation. And so there's 3 forms of radiation: ionizing, UV, and non-ionizing. Now, all forms of radiation cause damage, and their general problem with what the damage they cause is free radical formation. They particularly affect DNA. So that's really remember, free radicals could do a number of different things, but the DNA is the big thing that's involved with radiation. When I talk about radiation, understand that what we're looking at is a photon or particle of radiation energy. So that's what we're dealing with. Ionizing radiation is the most severe. It literally knocks electrons out of their orbit, so therefore you make the free radicals. It has cumulative effects, so even a little exposure over a long period of time can cause major damage. It can cause immediate cell death. It interrupts cell replication, and it's very much involved in genetic mutations. Ionizing radiation will cause cells to swell, disrupts the mitochondria, changes the nucleus. It can actually turn tissue into more fibrotic tissue that is no longer even able to function. So that's actually, by the way, if I have any radiation therapy students in here, that's what we use to treat cancer. So it's very specifically pointed at particular cells to destroy them. So we can destroy cancer with radiation. We must be very careful not to destroy our natural cells. UV radiation is what we get from the sun. It's not nearly as severe. It does disrupt intracellular bonds, and so its damage really depends upon 3 things: what kind of UV rays, how intense is the exposure, and how much melanin pigment is actually in the skin. Fairer skinned people are more vulnerable to UV damage. That doesn't mean that people with darker skin are not. It's just that they're less vulnerable. They can have longer exposure, but they can still do damage, absolutely. And the non-ionizing radiation is radiation below those of visible light. So we're looking at infrared light, microwaves, and arguably even things like ultrasound can figure into non-ionizing radiation. That's more like older ultrasound machines and not what you're seeing currently. Some lasers and even older TVs could have non-ionizing radiation. They just cause the atoms to kind of vibrate and rotate a little, and then that's kind of how a microwave works. So they do increase thermal energy. They very rarely cause any kind of damage, but theoretically they can. Then you have chemical injury. So here's some of the major chemicals. Carbon tetrachloride, I have this one listed here because that's what's used in dry cleaning solvents, refrigerants, and some pesticides have the carbon tetrachloride in it. They can cause some reversible injury, like they are involved in some fat accumulation in some cells, but they also can release a free radical. Instead of the CCl4, it can be CCl3, and that's a free radical. It primarily targets plasma membranes and the endoplasmic reticulum membranes, and it's most damaging in the liver and the brain. Acetaminophen, everybody knows what acetaminophen is. It's Tylenol. It can cause necrosis of hepatic tissues, or the inflammation and death of your cells of your liver. It does cause free radicals to release, and it damages mitochondrial proteins. So this is one of those you have to accumulate over time. It's not like, oh gosh, I should never take Tylenol again. But if you're taking Tylenol regularly over a long period of time, that can be very damaging to the liver. Alcohol, I think you're probably all familiar with the kind of damage that can come from alcohol, but let me point out that alcohol can damage the liver, which you may already know leads to fatty liver disease, leads to free radical release. It can damage the GI tract. It's associated with carcinomas or cancers of the esophagus, the pharynx, and the oral cavity. It can cause pancreatitis and gastritis, so it's damaging to the whole of the GI tract. It causes brain damage, and not just while you're intoxicated, but prolonged use actually leads to degenerative brain disease. It can also lead to peripheral neuropathies, and what that means is that neuropathies are damage to the nerves. Peripheral nerves become damaged, so they lose senses of touch in the fingers, hands, feet. When you lose the sense of neurons there, or the neurons are lost and the sense of touch are lost, that starts to lead to problems with responding to injuries. Blood flow is diminished, and that can lead to necrosis in those limbs. It also can cause muscle damage. It can damage the heart when we talk about alcohol with cardiac issues. It causes testicular atrophy. You know what atrophy now means, so men should be aware of what that does. It leads to spontaneous abortions, and of course it also can be involved in fetal alcohol syndrome. So alcohol is a nasty one. And then we have lead. Lead is found in all sorts of things still in our homes, some paints, especially older homes. It can be in the soil, in dust, some cosmetics, pottery glazes, and industrial areas, especially where there's been a lot of welding. It does cross the placenta. It can lead to birth defects, and in large doses can cause spontaneous abortions as well. It is absorbed through the GI tract, or it can be inhaled, particularly if it's in the soil, it's a dust. You can breathe it in. It does lead to nutritional deficiencies. It interferes with calcium, iron, and zinc absorption. So we're going to talk about the GI tract in a second, but lead actually binds to places where calcium would normally bind, or where iron would normally bind, and therefore it does not allow you to bind to those other elements or other minerals, and they're lost. Even if you take them in and eat them, they come out in your waste. You don't hold on to them because the lead is in the way binding to them. It can be stored in our bones, blood, soft tissues, and it does cross the blood-brain barrier, particularly in infants and children. So what you actually see happening is that you will see that some kids are diagnosed with things like ADD, and what they really have is lead poisoning. The actions of lead: it inactivates enzymes, interferes with calcium, as I mentioned already, particularly in the bone, and it interferes with nerve transmission and brain development. 4 major areas of the body. Be sure you read this section in the chapter. Lead always shows up on our exams because it's really still a common problem, particularly in the Mid-South, so you'll see it a fair amount. In the nervous system, it damages both the peripheral and the central nervous system. So I had already mentioned with kids the ADD, but for adults, it actually allows too much neurotransmitter to be released. Leads to hyperactivity, seizures, convulsions, delirium, and coma. So again, in the nervous system, In the central nervous system, it is going to affect the brain with neurotransmitters, hyperactivity, seizures, convulsions, delirium, and coma. In the peripheral nervous system, it leads to paralysis, particularly in the hands, feet, and wrists. In the blood, it specifically targets the red bone marrow, preventing the formation of hemoglobin. So that inhibits the enzymes. It leads to anemia. It also targets existing red blood cells, binding to it and in preventing it from binding to oxygen, so it leads to hypoxia. In the GI tract, this is the main problem with adults, is they have what's referred to as lead colic, C-O-L-I-C. You've heard of a colicky baby. Well, in this case, lead colic is a very severe but indistinct form of acute abdominal pain. By indistinct, what I basically mean is there's pain. Can't exactly point to where it's coming from, but it's severe. That's lead colic. And then it also damages the tubules of the nephron at the kidneys, leading to renal failure. And what winds up happening is you no longer hold on to your glucose, your phosphates, and your amino acids. All those things start going out in your urine, and that's, you can test for it. If you find high levels of phosphate, amino acids, or glucose in the urine, you rule, for instance, in the glucose, you rule out diabetes, then you want to start looking for lead poisoning. So please read that section, it's important. And then finally is a little bit on necrosis and gangrene. Take a look at pages 44 and 45. Your book does a good job of explaining that. Do take a look at what the difference is between the types of necrosis and gangrene. Necrosis is dead tissue. We're looking a little more when we talk cellular level, when we talk about this. And then there are 3 forms of gangrene. That's more of a broader, more generic sense of terms. So they're very much related to each other. But know the different types and what the characteristics are. For instance, the gas gangrene has to do with the bacterium Clostridium, whereas dry gangrene has to do with lack of blood flow. Wet gangrene has to do with backing up of blood flow. So take a look at those. And then finally, just a word on cell injury. Cellular aging. At the very end of the chapter, there is a little section on genetics and aging. Talks about how when DNA replicate, the tail end of the DNA, which is called the telomere, is not replicated, but it's attached to the end of the DNA. So it cuts when they're dividing, and that telomere lists at the end, and then it's reattached. And that reattachment occurs by the enzyme telomerase. So as cells age, it appears telomerase diminishes. So I bring this up now, not that that's like if we could replace telomerase in the body, no one would get old. I bring it up mostly for you to understand that that's what happens as cells age or wear out. But cancer cells tend to have an enormous amount of telomerase. So when we do the chapter on cancer and the lecture on cancer, I want you to be thinking about, hey, we talked about that as cells age and they're ready to be recycled or getting old. The telomerase, that enzyme, leaves, and so we lose part of the DNA and the cell starts to degrade. Cancer cells don't have that problem, which is why they can continue to sustain themselves. So just read that little bit about that at the end of the chapter as well. And that takes us to the end.